back to Histology 101 live stream. We're going to continue our look now at the histology of the liver, the gallbladder and the pancreas. So we'll just jump in here. So this is a section of the liver from a pig. Now we can tell that it's a section of the liver of the pig because actually it has these very well defined lobules. So the liver is made up of lobes, 4 lobes, right, left, hepatic and caudate lobes, and then those lobes are subdivided by connective tissue or stroma into lobules, and in the pig the stroma is very defined. It really defines the structure of the lobules, whereas humans, and I'll show you a quick section of human liver here, we can't see as a well delineated stroma, and therefore the lobules are much more difficult to define. So we're going to start off by having a look at a section of the pig liver, because that actually helps us think about the actual structure of the lobule within the liver, and then we can actually come back and have a look at this if time permits. And if you've got any specific questions about that. Whoops, wrong way. Right. So as I say, it's made up into lobules. There's a number of ways we can actually classify the liver histology. The classic sort of histologist way is to think about a lobule like this, where we've actually got points. We actually sort of roughly describe them as being made up of a hexagon shape, which is 6-sided, although 1, 2, 3, 4, 5, 6. You can sort of see it's not quite a hexagon, but it's roughly a hexagon. So we think about it, it's characterized by having this little vein in the center here. This is known as the central vein, and then we've got a blood supply at each of these little regions here, these little points of our hexagon. And we have sort of what we call a dual blood supply in this region, and the blood sort of coalesces and pools and then eventually drains into this central vein. So as it does, the hepatocytes, which are the main functional cells within the liver, or the parenchymal cells, they then sample that blood, they add their liver proteins to it, the plasma proteins that they make, and then they actually take out the nutrients, get rid of toxins and so on. So we know that the blood supply within the gastrointestinal system is that the intestines take all the reabsorbed digested material. It then goes to the liver via the portal vein before it eventually goes back into the circulation. And that way the liver actually samples that material. Now these points of the lobule are referred to as portal regions or the portal triads because they contain 3 different components. They contain what's known as a branch of the hepatic artery. So this is actually a branch of the abdominal aorta, and it carries high oxygenated blood. Oops, sorry, I'm doing this with the mouse. I'm not actually using the thing there. High oxygen blood and moderate levels of nutrition. We've got the, let's go blue here, a branch of the hepatic portal vein. Oh, that's a bad P. That actually has low oxygen, but actually has high nutrient levels because it's actually taken all the nutrients from the gastrointestinal tract. And the third structure that we identify in this region here is the bile duct. Whoa, that was a big B. And so the bile duct is actually receiving the bile from the hepatocytes. So instead of the blood flow sort of going from the outside to the inside to the central vein here, the bile flow is actually going the opposite way. So it's actually coming out to these portal regions, to the bile ducts which exist within these regions. So this is what we call in sort of histology terms the classic lobule structure of the liver, where we've got our hexagonal shape with the points of the hexagon actually containing the portal regions or the portal triads containing branches of these 3 things: a branch of the hepatic artery, a branch of the hepatic portal vein, and a branch of the bile ducts or a bile duct. So here at these points in the sort of classic histology model, or the classic histology lobule, the blood coming from the hepatic artery, the portal vein, coalesce, pull together, and then travel through sinusoids down towards the central vein here. So we're going to get rid of this. We'll close this and we can actually see some of these structures here. So we can see this tissue out here. This is the stroma, so this is connective tissue. We can see a blood vessel here. Okay, so this is probably going to be one of the branches of the hepatic portal vein. The hepatic artery, not always obvious in every section, but we're looking at more like something like this, where we can actually see smooth muscle within the wall. So we often say, there's the hepatic artery, we have the hepatic portal vein and the bile duct in those portal regions, but in fact they're the smaller blood vessels of those. So the hepatic arteriole or hepatic portal venule, for example. So this is probably a hepatic arteriole carrying the high oxygen blood. This structure, you can see the nice round nuclei here. This is actually the bile duct. So bile ducts are lined by a simple cuboidal epithelium. So that's what we're seeing here. So here's our bile duct, and that would be receiving the bile from the hepatocytes. Again, we've got the bile duct. See nice round cells for the simple cuboidal epithelium. And we should just sort of zoom around and we can take a look at the stroma here, and every now and then we can actually see like the branch of the hepatic portal vein here. This is actually probably a valve leaflet for this venule here. So the veins, of course, tend to be thinner walled and larger lumen, whereas the arterioles tend to be the smaller ones with the muscle in the wall. And again, we're looking at things like this, which is the bile duct in this region, whereas this is more the hepatic portal vein, or the branch of. All right, so here's our central vein here. So this space is the central vein. You can see it's lined by these flattened endothelial cells. So that's receiving the nutrients through these sinusoids. So this is a sinusoid. You can see the red blood cells within the sinusoid here. Here's another part of the sinusoid. So the blood starts off in the periphery here, and then pulls together and migrates through the sinusoids, down the blood vessels, and eventually draining into this central vein here. And eventually that goes back to the hepatic vein, back to the inferior vena cava, and back into circulation. So you can see the endothelial lining here. So these are actually the simple squamous endothelial cells of the sinusoids. And then these cells here with the round nuclei and the pseudophilic cytoplasm, these are the hepatocytes. And they form these kind of cord-like structures on either side of the blood vessels. So they actually have this endothelial lining just above the surface. Now we don't see this with light microscopy, but the sinusoidal endothelium is actually an incomplete endothelial layer. So there's big holes between the cells that enables quite free access for the blood to the hepatocytes. And the endothelial cells sort of sit just above the surface of the hepatocytes, so that the hepatocytes can actually form microvilli, so that they can increase surface area for absorption. And so there's a little space there between the endothelial cell and the hepatocytes, and that space is known as the space of Disse, D-I-S-S-E. And it just is a way in which the hepatocytes can form microvilli and interact with the blood vessels, and allows diffusion and reabsorption and things like that to take place. The space of Disse is also thought to be the origin for lymph. Now, lymph is, of course, extracellular fluid, and so it's thought that the lymph sort of runs through the space of Disse and goes back out to those portal regions where we find a lymphatic vessel out there. So when we talk about portal triads, we're really talking about portal, what's the four? Quads? Quads, yeah, anyway, you get four things. There's four things out there, but we just call them portal triads. Yeah, so these are the hepatocytes, the nice round nuclei. They've got the little prominent nucleolus, and then the various pseudophilic cytoplasm. Now, the darker cells which we see are either going to be part of the endothelial lining. So these are the endothelial cells. The more darker stained, rounder ones are going to be one of two cell types. They're either going to be Kuppfer cells, which are the resident macrophages within the liver, and they sort of hang around the sinusoids, ready to phagocytose any invading microorganisms, also getting rid of aged red blood cells, so the liver can function to help remove old red blood cells as well, and the Kuppfer cells play that role. The third cell type which may exist within the liver and may be found associated with the endothelial lining is known as the hepatic stellate cell, or the cell of Ito. Now, they're thought to play a role in synthesis of vitamin A, so they have lipid droplets associated with that. But they're also thought to be reactive during pathologies of the liver. So whenever you get fibrosis of the liver, it's thought that the hepatic stellate cells actually undergo hypertrophy and potentially hyperplasia, and therefore increase in number and cause a deposition of increased connective tissue. So anything sort of rounded, we would say, is either a hepatocyte, is either a Kuppfer cell or a hepatic stellate cell, or a cell of Ito. Okay, so that's pretty much the sort of classic lobule structure of the liver. So as I mentioned, the blood comes from the periphery, percolates in, is collected by the central vein here, and the hepatocytes deal with the nutrients or toxins that are coming in. Now, there are two other ways to actually think about the structure of the liver in terms of the liver lobule structure. Now, one of these actually sort of, we'll get my little drawing thing here, sort of thinks about connecting three lobules. We'll sort of do a rough diagram here, where you actually sort of connect the central veins like this. And you can see that the center of attention here is actually one of these portal regions. So all of the cells within this region are actually creating bile, and the bile that's being produced within these regions is flowing back to the bile ducts within this region. Who remembers the name of this model? Who remembers the name of this type of way to think about the liver structure? The sort of triangle one. No one? I'm just waiting for the delay. There's a little bit of a delay in the stream. The portal lobule, yeah, so that's the portal lobule. That's the way we can actually think about it. And so it considers bile flow. So instead of thinking about the blood supply flowing into the center here, it's thinking about all of the cells within these zones contributing bile and flowing back towards the bile ducts in the middle of these regions here. So here we've got our bile duct, and this will be collecting all the bile from these hepatocytes. The third way to think about the blood supply, the third way to think about the liver structure is the most clinically relevant. And so previously, whoops, wrong button. Previously, when we talked about blood flow, we talked about it arising from these points and then flowing into these central veins here. But in fact, what we saw when we zoomed in here, we did see blood vessels within all of this stroma. So in fact, blood supply actually runs in this way. So it actually diffuses or percolates through, or perfuses is probably the better term, through the sinusoids from the stroma towards the central veins here. And what this does is it actually divides the liver structure into zones. So here's zone 1, here's zone 2, and here's zone 3. And then on this side, here's zone 1, here's zone 2, and here's zone 3. So here we're actually thinking about the cells which are dealing with the blood supply that's percolating through, diffusing, my terminology, I've lost it in the afternoon here, perfusing through these regions from the stroma back out towards the central vein here. And so cells in zone 1 receive the blood supply first, then cells in zone 2, and then cells in zone 3. So clinically, if we think about someone with hypoxia, for example, right-sided heart failure, for example, where there's low oxygen, the cells in zone 1 are actually going to take up most of that oxygen. Cells in zone 2 are going to have less oxygen to actually deal with, whereas cells in zone 3 are going to have virtually no oxygen. And often we'll see is someone with hypoxia is actually cells within zone 3 undergoing necrosis. They're actually undergoing cell death because they're not getting enough oxygen supply. Now, the opposite can be thought of as well, as if someone actually takes an overdose of a toxin, for example. Then cells in zone 1 are going to deal with that toxin first and therefore be most affected. Cells in zone 2 moderately affected, and then cells in zone 3 being least affected. So this is, as I say, the most clinically relevant way of thinking about the liver structure, and this is described as the liver acinus model. So where we get zones of cells which are dealing with blood supply or perfusion of blood. Now, does that make sense to everyone? Thank you, by the way, Red Cherry Waves, for contributing the answer. It's 3:50 in the afternoon. I probably haven't had enough coffee, so you really helped me out there, because I asked the question because I couldn't think of it straight away. So, but I hit that well, I think. I don't know if anyone really noticed that. So that's, as I say, the liver acinus model of thinking about the liver structure. So hopefully that makes sense. So we've got those three ways. The histologist way, thinking about the classic lobule structure. We've got the portal lobule structure, thinking about the central region of the portal region being the center of a triangle, and all the cells within that triangle forming the bile, and it's being secreted into those bile ducts there. And then this liver acinus model thinks about the blood supply, the perfusion of blood, and how these cells deal with that blood. All right, so any questions about the liver there? As I say, this really shows up some lovely features of the stroma versus parenchyma, the functional parts. We might see these little regions here. We can actually see these accumulations of blood cells, white blood cells. So we've got some sort of inflammatory process taking place here. I'm not exactly sure which, I'm not a pathologist, but you may have noticed these in your sections if you were doing them, for example. So here, what do we think this is? What's this structure here? Right, so I've got, let's use this little label. We'll do this. We've got structure 1, we've got structure 2, and here we actually have structure 3. All right, so does anyone want to hazard a guess about what structure 1 is? All 6 of you, who I can see is actually watching at the moment. Yeah, good, Ruby, well done. That's the bile duct, and we can tell it's the bile duct because it's lined by a simple cuboidal epithelium. Nice round nuclei, typical of a cuboidal type of epithelium. Well done, Ruby. Number 2. Always got to wait for the little delay in the stream. But if no one's going to answer, I can answer. So this is actually portal vein. Yes, Dr. Shio, thank you very much. Yeah, so this is a branch of the hepatic portal vein. So we can see big lumen, irregular shaped lumen and very thin wall comparatively. Number 3 then, it's going to be pretty obvious if we think about a portal triad. There's another branch of it up here too. So number 3 then is the branch of the hepatic artery. So this is the hepatic arteriole. So these are the smooth muscle cells here on the wall. You can see it's quite a small lumen compared to the hepatic portal vein branch. There's another little branch of it up here as well. That's just sort of, beginning our interpretation of, these regions here. Again, lots and lots of white blood cells here. Here we've got the bile duct. So if you sort of scan along, we can actually see these different things. Again, we've got the vein here. Here's the bile duct. Again, we've got the vein. Nice bile duct here. You can see this nice, looks a little bit like a guitar. All right. So, this is a section of the liver, and prior to the death of this animal, I think it was a rat, they injected the blood with, injected the rat with India ink. And India ink has a predominance of carbon within it. And so wherever the carbon is, you have these very black regions. And so what they did was they injected the rat with the India ink, they let it circulate for a while, and then obviously they euthanized the animal. No worries. Thanks, Dr. Shio. Thanks for popping by. I really appreciate you coming in and contributing to the stream. Good luck with the conference. I hope it goes well. The India ink then appears as these very black regions where cells have actually taken up that India ink or the carbon. And of course, we think about the cell types within the liver. The cells which are going to take up that carbon, of course, are going to be the resident macrophages, the phagocytes within the liver, and those are the Kuppfer cells. So here we've stained the section with H&E, and wherever there's black, that means there's cells which have actually phagocytosed that carbon material. So here, for example, here we've got our central vein, and then all of these black regions are the regions where we actually find our Kuppfer cells, the resident macrophages. So we can see them sort of radiating around this central vein. Here's the main body of the Kuppfer cell, and you can see these little cytoplasmic processes that are sort of extending out as well. So it really shows the sort of distribution of Kuppfer cells within this tissue. What you might also notice of the hepatocytes are they look like they've got holes within their cytoplasm. So they've got these nice round nuclei, prominent nucleolus, which is really typical of a hepatocyte, but the acidophilic cytoplasm has these little white spaces. Now, who wants to hazard a guess what the white spaces are within these cells? Are they artifact, or is something else going on? Glycogen, Ruby. Ruby's on fire. Someone get the fire extinguisher. Ruby's on fire. Well done, it is glycogen. So the fact that these cells receive the nutrients, they're actually able to store the glucose in the form of glycogen, and glycogen is an inert substance, so it doesn't actually interact with either hematoxylin or eosin. So if we actually go to the next slide, this is actually a way to stain for glycogen. This is the PAS stain. So PAS stands for periodic acid shift stain. And what happens is you first incubate your section in periodic acid, and what that does is it converts glycogen into an aldehyde, and then we use our shifts reagent to stain the presence of those aldehydes. So it actually is a way to actually determine where glycogen is within cells. So this is a section of the liver being stained with this PAS, and we can actually see the nuclei. But this very magenta stain is where we can actually see the glycogen within the cytoplasm. So in that previous slide where we had the holes, and we go back to this PAS stain, this is where the glycogen is. So this is sort of almost a confirmation of showing where the glycogen is distributed within this cytoplasm of these hepatocytes. These spaces are, of course, where the sinusoids are. So that's one of the classic features of hepatocytes. They store glucose in the form of glycogen, and they often appear as having these holes within their cytoplasm. We didn't see that in the previous slide very much at all, but we can see a little bit of evidence of the holes within the cytoplasm here. All right. Does anyone have any questions about the liver before we begin our look at the gallbladder?
So I do.
Yeah.
So you said in the pig that the stroma is quite well defined and it helps you see the structure. So when confronted with the human liver, what's the decision tree that you would go through to decide that you were in the liver? Is that something that you can talk about?
Yeah, so we've actually got this section which we never came back to. So this is a section of the liver here through the human, and we can actually see, is there a use for the portal lobule module? Isabella, use, not really. It's just another way to think about the liver structure in terms of function. So the classic lobule structure is the histologist way to think about the structure. The portal lobule way is a functional way to think about the liver structure in terms of bile flow or potentially for lymph flow. And then the liver acinus way is the more clinically relevant way to think about perfusion, blood supply. So there's really not much use other than a sort of a functional way to think about the liver structure. So getting to James's question, so this is the human liver. You can see we can get a little bit of idea about where some of the stroma is, but we don't have that lovely defining lobule-like structure that we see. What we look for when we're looking at a section, the cords are usually really well defined for the hepatocytes. So we've got these lines or these cords of cells of hepatocytes with the white spaces in between them. So that's what I'd be looking for. So I don't know if did everyone hear James's question about when we're looking at a section of the human liver, the fact that it doesn't have those lovely delineating stroma for the lobules, how do we tell we're actually looking at a section of the liver? What's our decision tree, for example? So what we're looking for are these lines of cells with the nice round nuclei centrally located. Often we have the prominent nucleolus present, acidophilic cytoplasm, and then these quite large spaces in between. And these, of course, are the sinusoids between these hepatocyte cords. So if we zoom around, we can actually see it's a little bit more. Sorry. Right. Thank you. I just had to time out someone, sorry. Someone's been a bit of a dick in the chat there, to use the anatomically correct term. We see we don't have that lovely delineation like we saw with the connective tissue stroma. What we do see are these things like here, these are actually the bile ducts. So it's a little bit more complicated. So what we're looking for, as I say, are these cords of cells, nice round nuclei, only one cell layer, generally, and then these wide spaces, which are, of course, the sinusoids. You can see here's a central region, but if we zoom out, it doesn't have that nice sort of where are the portal triad regions. So roughly they're in these sort of regions here, and they can be a lot more difficult to interpret. Generally, though, if I'm going to ask a question in an exam, it's going to be quite easy to distinguish that you're looking at a section of the liver. So I'm not there to try to trip people up. But it's a very, very, I think it looks a bit like a maze. This is the sort of tissue that I look for. It looks a little bit like a maze with the holes there. And we've got these sinusoids filling these spaces between these cords of cells. So it's quite maze-like in appearance. So I don't know, does that sort of...
Yeah, I just thought it might be interesting for people to think about the human stuff. I know
Yeah.
Rosalind was talking about it
Yeah.
in the pathology shoot last week.
Yeah, and I alluded to the slide of the human and I never got back to it, so thanks for reminding me. Again, we've got one of these regions with the bile duct and the portal vein here as well. I can't see if I can see the little arteriole there. Maybe this structure here, you can see the smooth muscle. Maybe a little one down there, but the bile ducts are quite obvious compared to the hepatic portal vein branch. All right, any other questions about the liver? We can move on and look at that section of the liver and gallbladder. All right, so this is a section of the liver and gallbladder. It's got these really nice sort of arrangements here in the central canal. The cells aren't particularly well preserved, unfortunately, but in fact they look quite foamy in appearance here. So here's our central vein. We've got our portal regions out here. You can see the bile ducts, branch of the hepatic portal vein, some of the bile duct there. There's this one here. This one looks like a bit like a face. It's kind of grimacing that way. It looked like it was smiling from the other way, but now it's kind of grimacing. Anyway, so this here is the gallbladder. It's very thin-walled here, but it's characterized by having these sort of projections here and a simple columnar type of epithelial lining. Now, the gallbladder, of course, is responsible for absorption of water and concentration of bile. So often when these cells are actively absorbing the water, we end up with these spaces in the lateral sides of the cells. And that's because the cells are absorbing so much water that they can't move it down the osmotic gradient. And so rather than sort of, they sort of keep it laterally between the cells. So it has a mucosa of this simple columnar epithelial lining. There's no goblet cells present within the gallbladder. So this is one of the ways that you'll distinguish it from a section of the small intestine, for example. Often we have these structures which look a little bit like villi, not necessarily so much in this section here, but if I move to the next section, for example, right, we're going to flip this around. You can see this sort of villus-like structure I was talking about. So we've got this simple columnar epithelial lining with a core of loose connective tissue, and I suppose it may be possible to accidentally identify something like this as a villus. To me, this looks a little bit like a villus. The difference being there's no goblet cells, there's no brush border like we see. You'd see a lot more capillaries and a lot larger layer of loose connective tissue or lamina propria beneath. Whereas in the gallbladder, we have also these weird little tunnels that appear in this mucosal lining as well. So you can see them here. So if you see that sort of structure, then hopefully that should lead you towards identifying this as very irregular in appearance, aren't they? They look really odd. They look like someone's doing, rabbit fingers over here, like someone's doing that, and it looked like weird sculptures or something, or corals or something like that. Anyway, they're irregular in shape. They're not all the same height like we see in the small intestine with the villi and so on. And they, as I say, the thing to look for here is this sort of tunneling type of appearance that you can often see. Very classic of the gallbladder. It has a mucosa, it has a muscularis. It doesn't typically have a submucosa, although some textbooks do describe a submucosa, but you can see here, we're pretty much moving from the mucosa to the muscularis here. This is the smooth muscle of the muscularis. And then we're out to an adventitia, eventually and a serosa. We sort of zoom out. Not sure we can see any again, the textbooks are a little bit confused about whether there's serosa or an adventitia. Really, I suppose it really doesn't matter. But again, we're moving clearly from a mucosa to a muscularis, sort of bypassing the submucosa, and then we've got the blood vessels out here for the adventitia. All right. Any questions about the gallbladder here? So, do the tunnels in the epithelium have a name? They do have a name. It's a name in pathology. Anyone able to help me out? I can, I shouldn't, but I can Google it. I haven't referred to them, but they do have a name. It's again one of these eponymous names, so it's named after someone. It's quite a complicated name, and maybe you come back to me. I don't know if James can Google it on my phone. So Google tunnels gallbladder. They've got a complicated name. All right, so that's the gallbladder. We know that the smooth muscle is responsive to both CCK, or CCK in particular, so the cholecystokinin hormone that causes constriction of the gallbladder to move the bile out into the bile ducts and into the duodenum to help emulsify the fat. Of course, bile is about emulsifying fats, breaking them down into smaller components so that it can be dealt with by the pancreatic lipase and so on. All right then, so we're going to move on to the pancreas. Now the pancreas, if you remember, is a dual gland in the fact that it's both endocrine and exocrine portions. Now the endocrine portions exist as these pale staining regions here. This is a crypt of Leberkuhn or a pancreatic islet. Sorry, crypts of Leberkuhn. Islets of Langerhans or just pancreatic islets is the more common term for them. And these are where we find our alpha, beta, delta cells producing things like insulin, glucagon, somatostatin, and so on. The other part of the gland, of course, is the exocrine portion of the gland. And so it's predominantly made up of these acini structures, these acinar structures, secretory cells arranged into a circular arrangement, and they secrete into ducts. And here's an example of a duct here. That's sort of lucky. They're quite difficult to see in comparison to the salivary glands. If you remember when we looked at the salivary glands were a very prominent feature of the ducts were a very prominent feature of the salivary glands. Whereas here you can see it's very, very difficult to actually see the ducts at all. We can see the islets of Langerhans, but the ducts are much more difficult to visualize. You can see them here in the connective tissue. Can't find it?
Not yet.
Right. So here's our secretory cells. They often have this basal basophilia because these are cells which are producing the digestive enzymes. So they have lots of rough endoplasmic reticulum at the base of the cell, and then that causes the basophilic staining at the base of the cell. The top of the cell stores the secretory products in the form of secretory granules or zymogen granules, and then eventually they're exocytosed into the duct system itself. So in a nice stained section of the pancreas, we often get this basal basophilia and this apical acidophilia. Now, this section hasn't really stained like that. We do get a little bit of idea of the basal basophilia, but not that nice pink granule type that we actually would expect to see. Maybe the animal that this came from had recently eaten and had exocytosed its zymogen granules. Here's a little duct. So as I mentioned, it can be difficult to visualize these sort of things here. You can see how we get these cells here. Now the pancreas has this unique arrangement. So when we looked at the salivary glands, they had these ducts, and then they had the secretory acini sort of running this way. So here's our secretory cells. Okay, and we've got our nuclei of our acinar cells down the bottom here. And then we have our duct cells here, the intercalated duct cells. Okay, and so that's the sort of typical arrangement that we saw for the structure of the acini in the salivary glands. Now, the difference being in the pancreas, what happens is that some of these duct cells actually extend down into this region, and so the duct cells sort of extend down here. And these are known as centra acinar cells. So these are known as centra acinar cells. Okay, and so these are classic appearance of pancreatic acini, and they can be difficult to visualize. But actually, what we're looking at here is some of these centra acinar cells. Can you see these squamous cells here? They extend down. Here's the acinus. And then we've got these nuclei right in the middle here. These are some of the centra acinar cells of this intercalated duct. So the centra acinar cells are extensions of that intercalated duct within the acinus. And then eventually they would drain into this more classic appearance of this intercalated duct. So in the salivary glands, we had the intercalated duct draining the acinus, and then it moves to a striated duct. In the pancreas, we don't have striated ducts, so they move from the intercalated duct into what's, I suppose, an extension of the intercalated duct, which we call the intralobular duct. So here's an example of one here. So here's the cells, and so this is draining into this intralobular duct here, and then eventually they would drain into the larger duct systems in the periphery, like this structure here. So this is a big duct in the connective tissue here. And you can see them sort of emptying. These are the ones coming from the acini. So what you're looking for when you're trying to identify those are the acini, and you're looking for sort of cells which are located inside them. And they're not always that easy to see. And of course, I would never really ask you to identify those. There's some here. See the flattened nuclei? Again, we've got some over here as well. So that's part of the duct structure there. You know, these centra acinar cells are located here. So again, one of the ways to distinguish between the salivary glands and the pancreas is the lack of abundance of those ducts. They were really, really prominent in the salivary glands, if you remember. They had, connective tissue around them. They were very pale staining, very prominent features where you almost don't see them. You have to really zoom in and do a bit of searching to find the duct systems within the pancreas here. So again, if we just take a random region of the pancreas, do central acinar cells and acinar cells have different functions? Yeah. So acinar cells are responsible for the production of the enzymes. So they have the zymogen granules, and they're responsive to CCK. Whereas the central acinar cells, being duct cells, they're sort of extensions of the intercalated duct. They actually are responsible for the secretion of bicarbonate into the pancreatic juice. And so they're responsive to secretin, which is the other hormone typically released in response to food moving into the small intestine by those enteroendocrine cells. So yeah, they do have separate functions. Acini are responsible for secretion of the enzymes in response to the detection of amino acids within the small intestine and the release of CCK, cholecystokinin. And the intercalated duct cells are responsive to secretin in response to a drop in pH as that acidic chyme moves out of the stomach into the small intestine. What are the thin white spaces between the acinar cells? That's a little bit of shrinkage artifact, if that's what you're talking about, Ruby. So this is a little bit of shrinkage artifact that's taken place. When we process the microscopy, we dehydrate, everything shrinks a little bit. And so that's what those little spaces represent, if that's what you're talking about. Yeah, so here's a little pancreatic islet. Let's see if we can see another duct. As I say, they're not always that easy to see, and they can take a little bit of searching. Yes, that's what I'm referring to. Great, no worries. Glad I was able to point that out. It does look like little channels, doesn't it, a little bit between the cells, but yeah, it's just mainly artifact there. Well, as you can see, it's not always easy to find the ducts. They definitely don't stand out as much as like when we were looking at the salivary glands. There's part of the duct system here, although it's a bit difficult to interpret. I'll zoom out a little bit again. There's a little bit of duct there. Yeah, okay. Well, that pretty much... Oh, there's the duct, of course, just as I sort of stop looking. See this nice, nice simple cuboidal type of epithelial lining. You can see it's receiving the secretions through this duct here. All right. Does anyone have any questions or something from the past alimentary system that perhaps you'd like me to go over? I'm happy to go over anything again really quickly. We've got maybe 10 minutes to go. Otherwise, we can just sort of call it a day. And then we'll do another stream where we have a look at the urinary system. Specifically, spend some time looking at the kidney and the ureter and bladder and so on. All quiet on the chat front. All right, well, I might leave it there then. Hopefully that gave you some helpful tips in identifying the components of the liver, thinking about the structure of the liver, looking at the gallbladder, and thinking about how you can differentiate it from the small intestine, for example. I'm thinking Ashkovsky or Shivsky.
Yeah, so the only thing that's come up, but it's definitely more of a pathological thing, is this Rokanansky-Ashkov sinuses.
Sinuses, yeah.
But whether they're what you were pointing, I'm not sure.
Yeah.
They tend to be deep.
That's the term that I've heard before. Just a quick closing note I thought mentioning, there's low latency setting in Twitch dashboard worth setting. Doesn't default to low for some reason. All right. Thank you very much for that tip. I'll have a look at that. If you're referring to the sort of delay between audio and video, I'll need to check that out. So thanks very much for bringing that up. We're all obviously very new to the stream. Thanks very much, Infinite. If you'd like to share something with me, it'd be really helpful if you could point out something in an email or on Twitter or something. That'd be really helpful, because I'm not exactly sure how to set that up, but I'll make sure I watch a few YouTube videos to see if I can try to fix that for future streams. Thank you, Ruby. Thank you for everyone for coming along. I know for those of you who stuck around for the full 2 hours, it was a pretty long stream to be talking all the time about histology. So hopefully you found it useful. I appreciate everyone coming along, asking questions. As I say, keep an eye out for future streams. We'll be looking at the urinary system, definitely for the second years. We may have a stream looking at the development of bone for our first years as well. And if you've got other things that you would like me to review, please hit me up on Twitter or through email. I think there's links around the place if you'd like to do it. Anyway, thanks very much for coming along. I hope everyone has a great night. For those UTAS students who are following along, have a great mid-semester break next week. And we'll probably have a break next week for our live stream, and that way you can come back and we can get onto the urinary system and the other systems that we'll be beginning our look at. All right, so that's it for now. Thanks very much. We'll see you in the next stream. Hooroo!